My name is Brian Maas, and I work in the quantitative pharmacology and pharmacometrics group at Merck. Today, I'll be talking to you about how to use model-based meta-analysis for RSV monoclonal antibody development in infants and children. These are my disclosures. This talk is separated into two parts. The first part summarizes how dose selection has been traditionally conducted for monoclonal antibodies in the RSV space, and it also introduces model-based meta-analysis, or MBMA, as an alternative approach for modeling during monoclonal antibody development. The second part will show you how we used a developed MBMA for RSV to support dose selection for MK1654, a monoclonal antibody that's currently in phase 3 development for the prevention of RSV. So RSV, or respiratory syncytial virus, is the most common cause of bronchiolitis and lower respiratory tract infection in pediatrics. It represents a huge unmet medical need, especially in infants and children less than 2 years of age, where the highest risk for RSV is. Now, there are different approaches being studied to protect this high-risk population. The first is vaccination of mothers during pregnancy. And the second that we'll be talking about today is passive immunization of the child with a monoclonal antibody. This remains a promising approach for RSV prevention. I think especially now after the COVID-19 pandemic, we've seen the need for innovative approaches for active and passive vaccine development. So even though this case study presents MBMA being used in the context of RSV, these techniques can actually be applied pretty broadly across other diseases as well as modalities. So how has development of RSV monoclonal antibodies for infants been done in the past? Well, historically, it's been done based on viral challenge studies in cotton rat. So the figure on the bottom shows a schematic of a typical study design where you administer monoclonal antibody prophylaxis, you challenge with RSV, and then you measure viral load in lung and PK in the serum. And the figure on the bottom right shows how you might set your PK target in the clinic. So you essentially target the concentration that's needed to reduce viral load by a factor of 2 log 2 units after challenge. So the goal is to then select a clinical dose that keeps your infant above this PK target for the duration of the season. And overall, this approach has shown to be reasonably well translatable to the infant population. But today we'll be discussing an alternative approach for development, and that's called model-based meta-analysis, or MBMA. This is an analysis that incorporates different models to account for the effects of treatment, time, patient population, and other variables on an outcome. It incorporates the effect of dose by tying in our standard PK/PD models, and it could also include trial level covariate relationships to account for between trial differences and patient populations. And it's often developed using mean level external data that's collected through something like a systematic literature review. So this is a huge advantage because you're able to leverage the totality of available clinical information that's out there in the public domain. So today we're going to review how to use this type of modeling approach for dose selection for an experimental monoclonal antibody. So as far as using this approach, the real advantage is you can better understand the exposure response relationship early on in the clinical development program. And really our goal at the end of the day is to get from dose to clinical outcome. And in the absence of proof of concept data, you can kind of use MBMA to define that exposure response relationship. And The approach is really predicated on finding a good correlate of protection. In the case for RSV, that correlate is serum neutralizing antibody titer, or SN titer. It effectively serves as a measurement for RSV neutralization activity. And in the case of monoclonal antibodies, higher doses confer higher neutralization activity. So here, the panel on the left shows how we can use clinical data from early stage studies to understand that relationship between dose, concentration, and then your correlate of protection, in this case, serum neutralization titer. And then the panel on the top right shows actually how literature data can be used through an MBMA to define that relationship between neutralization titer and incidence or a clinical outcome. And really, you put these sources together to bridge the relationship between dose and clinical outcome. And all this can be done prior to any real efficacy studies or dose ranging studies, really with just published clinical data and early stage understanding of PK and your correlate of protection. Now, in addition to gaining an understanding of your dose response relationship pretty early on in the program, there are also some other advantages of MBMA, and that's because you're using published clinical data, you're really reducing the uncertainty in translating viral load in an animal to clinical outcomes in humans. MBMA is also considering the change of protection over time, which allows you to account for time-varying components like changes in seasonality, since RSV is a seasonal virus in most places. And lastly, MBMA provides you with a continuous protection curve rather than a single C-trough protection threshold, which might be derived through preclinical challenge studies. However, there are some challenges that come with it, notably because you're extracting data from published studies. These data are not typically uniformly reported, so you're going to be dealing with heterogeneous data types as well as missing data, which you may need to impute for modeling. So at a super high level, this slide lists the steps for building an MBMA for RSV. Really, you start with your clinical database. And in the case of external data, this means conducting some sort of literature review, extracting and categorizing that data. You can see on the bottom left an example of how RSV clinical endpoints across studies were categorized into discrete endpoint buckets so they could be accounted for in the model. In this case, disease severity ranges from asymptomatic disease to ICU admission, with each disease severity category being a subset of the previous one. Then in step 2, once you have your extracted data, you need to fill in the missing pieces. In this case, like I said, your correlate of protection or neutralization titer is really the primary input of your MBMA. So for each entry in your database, you're going to need that information. And since it's likely not reported in all studies, it'll need to be derived or imputed using what's known about the antibody. And the last step is model fitting. Once you have your complete data set, your categorized clinical endpoints, your neutralization profiles, you could fit your model and evaluate and control for covariates such as disease severity, population, etc. So you now have your developed MBMA, and this slide shows how we can use that model in a clinical trial simulation paradigm to inform something like dose selection. Like most trial simulations, you start in the top left with your virtual population. You then simulate your known PK or correlative protection titer profiles for your virtual population. And based on that, the MBMA can then predict the disease incidence rate for your treatment and placebo groups. And that can be used to calculate something like a relative risk reduction, which is a common endpoint or measure of drug efficacy in a late stage trial. Okay, so I mentioned this is a two-part study. Here's part two, where we're going to show the MBMA that was developed and how it was used to support phase 3 dose selection for an RSV antibody MK1654. So just briefly, I've mentioned MK1654. It's a neutralizing antibody that binds to a very conserved epitope on the RSV fusion protein, antigenic site 4. Um, and there's huge opportunity for half-life extended antibodies like MK1654 in terms of preventing RSV disease in infants. And as I mentioned before, historically dose selection for the clinic has been based on animal studies to approximate what concentrations may be therapeutic, but here we're going to guide dose selection using model-based meta-analysis. So this slide shows the RSV model-based meta-analysis that was developed based on published data. So in the figure on the left, you can see, our correlate of protection, neutralizing antibody titer on the x-axis, and then incidence of RSV on the y-axis. And each data point represents a study arm that's extracted from our review, where red points come from vaccine studies and blue points come from monoclonal antibody studies. And we fit this data using an MBMA model to establish that relationship between these two variables. You could see that model-fitted relationship shown by the purple line here, which really demonstrates that across studies, increasing levels of neutralization titer result in a lower incidence rate of disease. Having developed that model, we then validated it using 3 separate approaches. We compared it against cotton rat. We compared it against published monoclonal antibody trial results from a phase 3 study. And then in the final validation exercise, we compared model predictions to efficacy results from an in-house phase 2 challenge study using MK1654. This study was a double-blinded, placebo-controlled study where healthy adults were randomized to receive either 1654 at varying dose levels or placebo before challenge. And then following challenge, they were assayed for viral load in the nose and incidence rate of symptomatic RSV. So this table on the left shows the proportion of participants with symptomatic RSV infection after challenges. And here you can see doses of 200 milligrams resulted in a numerically lower percentage of participants with RSV, and that trend continued for doses higher than that. And on the right, we show kind of the results of that validation exercises, where we show generally good agreement between what was predicted by the model and what was observed in the challenge study. So then lastly, having validated that model, we used it to conduct clinical trial simulation to predict the efficacy of 1654 in a phase 3 study. And results from these simulations are provided here in the table, which show that MK1654 is predicted to have high efficacy against RSV in infants for a 5-month duration. And these efficacy predictions were used to guide our dose selection for our late-stage studies, and they provided confidence that MK1654 can really yield high efficacy for the prevention of RSV in our ongoing trial. So thank you for joining. I look forward to your questions in the panel session.